Somatic hybridization. Somatic hybridization is also called parasexual hybridization. It is also called protoplast diffusion culture. See children, here it is. We selected two parental plants in the somatic hybridization. From these two selected plants, desired characters are present. What are the desired characters are present in the elected or selected parental plants? One parental plant containing cultivated plant characters, some other parental plant containing wild characters, wild plant. Wild plant is selected and cultivated plants are selected. Between these two plants, the elected protoplasts are fused to form new hybrid cell by the protoplast diffusion. See parental plant A. From the parental plant A, we isolated protoplast by the methods of mechanical and enzymatic methods. In the same way, parental plant B, from parental plant B also protoplasts are collected by the mechanical method or enzymatic method. These two different species of protoplast, treatment with fusion chemicals, PEG, polyethylene glycol, like these chemicals are mainly helps in the fusion of protoplast. See, fusion of protoplast to form one new hybrid cell. That hybrid cells are transferred into the nutrient medium. On the nutrient medium, that hybrid cell, somatic hybrid cell or protoplast diffusion cell develop into callus. That callus, treated with growth hormones, they develop into plantlets. See, these plantlets are mixed characters of the parental plants. Mixed characters of the parental plants means what are the desired needed characters are present in these two parental plants. The two characters are as is passed to the next generation by the somatic hybridization or protoplast fusion culture. It is the main theme of the somatic hybridization or parasexual hybridization. Somatic hybridization or parasexual hybridization. Now, see here it is plant A, plant B, isolated protoplast. The protoplasts are mainly taken in the petri dish. They are fused, called protoplast diffusion. They are transferred into the nutrient medium. They develop into callus and they develop into plantlet. See, somatic hybridization mainly following tips. First, we know what is mean of protoplast fusion culture or somatic hybridization. Fusion of two protoplasts to form somatic hybrids. See, we know what is mean of protoplast. Below the cell wall, all parts are called protoplast. In that protoplast, plasma membrane, cytoplasm, nucleus, remaining all cell organelles are present. Here it is in this somatic hybridization or protoplast fusion culture, fusion of two protoplasts to form somatic hybrids. See, it is also called parasexual hybridization. See, parasexual hybridization means vegetative cells are selected from the parental plants. Vegetative cells are selected from the parental plants to form new hybrid cells. So it is also called parasexual hybridization. See, it is the main procedure of the somatic hybridization. We select plant cells, plant cell containing cell wall. Here it is, cell wall is removed by the macerozyme enzymes. They are also called cellulase, pectinase, hemicellulase. By the treating of these chemicals, the plant cell wall is degraded and we observed only protoplast. In this way, from the selected two parental plants, we collected two different types of protoplast. the two different types of protoplast, protoplast A and protoplast B. They are treated with PEG, polyethylene glycol chemical. They are mainly helps in, means these chemicals are mainly helps in fusion of two different protoplasts. See here it is fusion of protoplast to form new hybrid cells. See hybrid cells containing hybrid nucleus also, hybrid cytoplasm to form one somatic hybrid cells. These somatic hybrid cells are cultured on the nutrient medium. We can produce the new hybrid plants. That hybrid plants are containing two parental characters. So what are the steps in this somatic hybridization? We'll discuss now. See here it is in this somatic hybridization or parasexual hybridization or protoplast fusion culture. The first step is protoplast isolation. And second step is protoplast fusion. See, third step is identification of somatic hybrids. Identification of somatic hybrids. And fourth step is post-fusion events. These are the main steps in the somatic hybridization or in the parasexual hybridization. The first step we know, protoplast isolation. And second step is protoplast fusion. Third step is identification of somatic hybrids. And fourth step is post-fusion events. So now first step is protoplast isolation. See the protoplast isolation collected from the leaf, pollen grains, callus. See protoplast isolated from the leaf, sometimes shoot, sometimes roots, pollen grains and callus. These are the main sources for the isolation of protoplast from the elected parental plants. Now, what are the steps? Isolation of protoplast mainly done by the two methods. One is mechanical method, second one is enzymatic method. First method is mechanical method, second method is enzymatic method, mechanical method after enzymatic method. And first step is we discussed the mechanical method. See children, in mechanical method, cutting the leaf, cutting the cell walls under a microscope using microsepals or sharp razors. And this process or this procedure to take more time to this mechanical method. See in this diagram, it is the peel or some cells of the elected parental plants. The elected parental plants containing cell wall with cytoplasm, protoplast, all are surrounded through cell walls, plasma layer, nucleus. They are selected from the parental plant. These parts are selected from the parental plants. See, these are observed under the microscope. We used sharp razors, microsepals, and slowly remove the cell wall. And finally we identified or we selected or isolated protoplast from the elected plant cells. See, protoplasts are taken in one petri dish after collecting of the mechanical method. See, it is take more time. See, it is the more time means slowly and patiently we identified which cells are under the microscopes. The cell wall is slowly removed by the sharp razors, microsepals. After removing of cell wall, we identified protoplast. We isolated protoplast. That protoplasts are selected from that parts and they are taken in the petri dish. It would take more time and we collected a less amount of protoplast, and also sometimes that protoplasts are broken protoplasts also collected. Broken protoplast means during the collecting of protoplast with using of sharp razors, microsepals, that microsepals and sharp razors also cut the plasma membrane sometimes. So that protoplasts are cannot utilized in the protoplast fusion culture. Only healthy protoplasts are collected. That healthy protoplasts are taken in the petri dish. It is the mechanical method for the isolating of protoplast from the parental plants. And the next second one important method is enzymatic method. In this enzymatic method, we used enzymes. that enzymes are called macerozymes. The macerozymes enzymes are also mixed with cellulase, hemicellulase, pectinase. These are used for the degrading of cell wall in the elected parental plants, and these are available in market, commercially available in our local markets, and they are isolated mostly from the fungi. That macerozymes are isolated from the fungi only. Now, what are the steps in this? See, children, we selected some plant cells. The plant cells are treated with enzymes. The plant cells are treated with enzymes like pectinase, cellulase, hemicellulase. By these using of these enzymes, the cell walls automatically degraded. Cell walls are automatically degraded. That cell walls degrading protoplasts are isolated in a Petri plate. See, cell wall is removed. Cell wall is degraded by the adding of the macerozyme enzymes. In this enzymatic method, we isolated large quantity of the protoplast. See that all protoplasts are in healthy condition. All protoplasts are in healthy conditions. Finally, we identified or we isolated protoplast. That protoplasts are taken in the Petri dish. See, it is the main theme of the enzymatic method for the isolation of protoplast. And steps in the method. See, surface sterilization of leaves with 70% of ethyl alcohol. See, which part is used for the selection of protoplast? That parts, not only leaves, the surface sterilization of leaves, pollen grains, callus treat with 70% of ethyl alcohol solution. Latter, washing with leaves with distilled water. Not only leaves, root, shoot, meristems, pollen grains, callus. They are washed with distilled water. And peeling of leaf pieces. Peeling of leaf pieces from the lower epidermis. See, here it is leaf pieces, sometimes root pieces, shoot pieces also collected. They are from the lower epidermis or surface of the epidermis. And that leaf pieces or pieces transfer into the kept in a 30% of mannitol solution, 13% of mannitol solution for the plasmolysis. For the plasmolysis, taken on the shaken water bath, it is mixed, means the mannitol solution always is in shaken condition. It is helps for the plasmolysis of the elected pieces from the plants. And placing of leaf pieces or leaf peels kept in cell wall degrading enzyme. Cell wall degrading enzyme mixture. The cell wall degrading enzyme mixtures are also called macerozymes. That macerozymes are mainly helps in degrading of cell wall. Finally, we mixed with the leaf peels or some peels of the elected parental plants. They are incubated for 24 hours at 25 degrees Celsius temperature. After 24 hours, we observed only we identified protoplast in that mixture, and the cell wall is degraded. Cell wall is degraded on these cells. Finally, that mixture, that above mixture is filtered by the centrifugation process, and separation wash with sorbitol. Separation and wash with sorbitol solution and placing in the 20% of sucrose solution. After that, pipette out protoplast after centrifuged and placing in culture medium. See, it is the main procedure. First, initially, that elected parts from the parental plants clean with 70% of ethyl alcohol. Latter, clean with distilled water. We collected peels from the pots and they are treated with the 13% of mannitol solution for the plasmolysis. Then after that mixture, the peels or pieces mixed with macerozymes enzymes at 24 hours at 25 degrees Celsius temperature, and they are filtered by the centrifuged and kept in the or washed with sorbitol solution and kept in the 20% of sucrose solution for the long storage. Finally, that protoplast from that sucrose solution, protoplasts are pipette out and protoplasts are transferred to the nutrient medium for the culturing. It is the procedure in the enzymatic method. In this somatic hybridization, widely used enzymatic methods only. By this enzymatic method, we collected large quantity of protoplast and at the same time healthy protoplast. Healthy protoplast also collected by the enzymatic method. Now, protoplast diffusion. It is the second step. In this protoplast diffusion can be classified into two types. One is they are spontaneous fusion of protoplast and induced fusion of protoplast. See, protoplast diffusion mainly two steps, two types: spontaneous fusion of protoplast and induced fusion of protoplast. Spontaneous means when we selected protoplast to different types of parental protoplast, that protoplasts are taken in the petri dish. Automatically or spontaneously, some protoplasts are fused without any induced method or without any signals to the protoplast for the fusion. That protoplast fusion is called spontaneous fusion of protoplast. And the second method is induced fusion of protoplast. See, in this protoplast fusion, we used chemicals for the fusion. Wantedly, we used some chemicals for the protoplast fusion. So this process is called induced fusion of protoplast. Induced means wantedly we used some chemicals for the protoplast fusion. So by these two methods, we collected different types of protoplast fusions. Spontaneous fusion of protoplast, induced fusion of protoplast are mainly used in the methods of the protoplast fusion. The next step is spontaneous fusion. We discussed here spontaneous fusion of protoplast. In this fusion, homokaryons are developed. See, homokaryons means fusion of protoplast done by the two methods or two steps. One is spontaneous fusion of protoplast. In this spontaneous fusion, only same species of protoplast are fused. Same species of protoplast means only one parental plant protoplast are fused. Here it is initially we selected two parental plants. From the two parental plants, two types of protoplasts are selected. In this spontaneous, only one plant protoplast are fused, called homokaryons. One parental plant protoplast fusion is called homokaryons. In spontaneous fusion, only homokaryons are developed. They cannot produce new variety of characters in the culture. So these are the disadvantages of the somatic hybridization. And the next step is induced fusion of protoplast. In induced fusion of protoplast, we use the chemicals. In these two types of sources, they are present. One is mechanical method, second one is chemical method. Mechanical method, we used pipettes, micro pipettes, and chemical method, we used chemicals. The chemicals are called fuse agents. And the fuse agents are available in our local market like SPVA, sodium polyvinyl alcohol, PEG, polyethylene glycol. These are the fuse agents. Including these fuse agents, lysozymes are also used as a fuse agents. See, we used chemicals. We used chemicals for the fuse agents, for the protoplast fusions. In induced method, we used micropipettes to the pressure on the protoplast is called mechanical method. And we used chemicals for the protoplast fusion as a chemicals is called fuse agents. That process is called chemical method of induced protoplast fusion. And we used chemicals or mechanical methods. This technique is known as electrofusion. Mostly we used chemicals for the induced method of protoplast diffusion. That process is called electrofusion. Electrofusion means we used chemicals for the protoplast fusion, and these are large scale fusion of protoplasts are improved. Means in induced method, mainly in that induced method for chemical method process. By the chemical method process, in this method, large scale of fusion of protoplasts are improved by the electrofusion or by the fusescence. See, the identification of somatic hybrids. After adding of these chemicals or we use pipettes or fusescents, we identify two types of somatic hybrids on the petri dish. One is homokaryons. Homokaryons we know already. Same species of protoplast fused, or same parental plant protoplasts are fusions that called homokaryons. These homokaryons cannot produce new variety of characters because that protoplast as it is produced the parental plant characters, not only new characters, only they can produce parental plant characters. That types of protoplast fusions are called homokaryons. And the second one is heterokaryons. In these heterokaryons, the protoplast fusion occurs between two different types of species. Two different species of protoplast fusions are called heterokaryons. In these heterokaryons, that somatic hybrid cells containing new variety of characters means the two different parental plant characters are present in the heterokaryon fusion of protoplast. So these heterokaryons are cultured on the nutrient medium. They are produced new variety of characters. The variety of characters are present or expressed by the heterokaryons only. Heterokaryons means different species of protoplast fusion is called heterokaryons. And the next and last one is post-fusion events. See, after fusion of protoplast, we selected which type of protoplast for the cultivation, for the culture. Here it is beginning, we identified, we selected what are the homokaryons, what are the heterokaryons. Homokaryons are cannot selected from that petri dish. Only heterokaryons are isolated, are selected, and that heterokaryons are transferred into the nutrient medium. They are developed into callus. Finally, that callus treated with the growth hormones, they develop into organogenesis. They develop into roots and shoots. So that plantlets containing good characters. See, these are the good characters of the plants. See, parental plant A containing some specific characters. Parental plant B also containing specific characters. From these two plants, we selected two protoplasts. They are fused to form homokaryons, including heterokaryons. From that karyons, we selected only heterokaryons. They are cultured on the nutrient medium. They develop into new plantlets. That plantlets are containing two parental plant characters. Here also two parental plant characters, mixed parental plant characters are present. So automatically these are developed into new variety of the characters. Example, see, some cultivated plants are cultivated by the human activities. We can provide fertigation, we can provide nutrition, we can provide irrigation also. But wild plant cannot provide fertigation, irrigation by the human beings. Means the wild plant resistant to the fertigation, resistance to the irrigation. These characters are mixed in the somatic hybrid plants. So these are the resistance to the water scarcity, resistance to the fertilizer scarcity, and they are also having good characters. It is the main aim and uses of the somatic hybridization.